The BA.5 Omicron variant is substantially more dangerous than previous COVID-19 variants. The scanning electron microscope image shows SARS-CoV-2 in yellow, also known as 2019 nCoV, the virus that causes COVID-19, isolated from a patient in the US, emerging from the surface of cells in blue and pink, cultured in the lab. The Greek alphabet is used to name COVID-19 variants. The World Health Organization, WHO, uses the Greek alphabet in consecutive order to name coronavirus variants. The Delta variant of COVID-19 is considered to be the most dominant. The following 8 variants named have been relatively insignificant. Nu was not utilized because it is easily confused with the word new. The letter Xi was not used since it is similar to a common surname such as that of President Xi Jinping of the People's Republic of China. As per WHO best practices, scientific names of diseases that can have political, racial, or geographic connotations and can be difficult to say, recall, or may cause offense are not used. Variants of concern. The BA.5 Omicron variant of COVID-19 is believed to pose a higher infection as well as morbidity and mortality risk to infected individuals than other variants. BA.5 Omicron is more dangerous than its predecessors since reinfections and severe outcomes are relatively common. The odds are higher of subsequent SARS-CoV-2 infection regardless of whether or not a patient was previously vaccinated. In a study of BA.2 and BA.5 Omicron vaccines as well as booster inoculations were equally effective against both Omicron variants. There was a somewhat higher rate of BA.5 reinfection in relation to same as compared with BA.2. For BA.5 infected patients, booster vaccination was associated with a respective 77% to 88% risk reduction of COVID-19 hospitalization and death. There was a higher 93% and 94% respective hospitalization and death risk reduction for BA.2 patients. BA.5 booster vaccination against COVID-19 confers substantial reduction of morbidity and mortality risk upon infection. The spike protein of the Omicron BA.5 coronavirus surface can attach to and damage cardiac muscle cells, triggering a damaging immune system attack. The SARS-CoV-2 spike protein interacts with other cardiac myocyte proteins, causing the corresponding inflammatory response. This toxic spike protein causes cardiac enlargement, inflammation, dysfunction, and cell death. Proteinaceous toll-like receptors, TLRs, mediate inflammatory pathways of the immune response towards a variety of pathogen-derived ligands, linking adaptive with innate immunity. Cardiac cell infection with SARS-CoV-2 activates TLR signaling. The SARS-CoV-2 spike proteins interact with toll-like receptor 4 proteins that recognize infectious pathogens, triggering corresponding inflammatory responses in cardiac cells, causative of morbidity and mortality. please refer to previously posted presentations on these subjects. Current expert consensus stipulates that especially individuals at high risk of severe disease who have not received the second COVID-19 booster should receive currently available recommended booster inoculation, even though they are not yet Omicron variant specific. In the presence of a current surge in Omicron variant infections and the absence of Omicron booster inoculation specificity, booster inoculation that is closest to the commonly circulated infectious version of COVID-19 is of value to everyone at risk. Given the currently available science, it is expected that Omicron-based booster inoculate will have a marginal impact in terms of disease prevention compared with currently available booster vaccines. The longer a person has gone without previously recommended booster vaccine inoculation, the person is protected less against infection and severe morbidity and mortality. Transmission electron micrograph of SARS-CoV-2 particles in gold within endosomes of a heavily infected nasal olfactory epithelial cell. New presentations coming soon. Please like, subscribe, share, and ring the bell for notifications. Thank you, subscribers.